Chapter 20, The Heart. Introduction. Now the purpose of this chapter is going to be to learn about all the components of the cardiovascular system. We're going to focus on the anatomy and physiology of the heart. We're going to review and discuss the cardiac cycle, and ultimately discuss all the various factors that influence heart rate and force of contraction. The first part is to identify the location of the heart. Now we look at this transverse section and we're looking from the underneath side looking up, and we notice that the heart is in a cavity, a specific cavity. Not only is it in the thoracic cavity, but we can elaborate a little further and say that it is in that mediastinum. So if you recall back to AP1 where we talked about all the different cavities of the body, we identified this secondary cavity in the thoracic cavity called the mediastinum. And recall that we said it was a rectangular shaped box going from the top of the sternum to about the diaphragm and then out onto the sides of the heart, which included the heart, all the major vessels of the heart, etc. We can look at an anterior view of the heart in the thoracic cavity. Again, if we were to take a pencil and draw a rectangular shaped box, we can outline the mediastinum, and we see a couple interesting things. The heart is lodged between the two lungs, but look at the lungs. Notice the right lung here has two fissures, which means it has 1, 2, 3 lobes. The left lung's a little bit different. There's only one fissure, which means it has only 2 lobes. So why? And the answer is, well, the heart. The heart is pushed down and off, angled to the left, and the lungs have to account for that. We see this cardiac notch inside the lung that the heart is going to sit in that left lung. We see that the apex of the heart, think of the heart, and we're going to erase everything we just did for a second because I want you to be able to understand the shape of the heart. So the heart is shaped like a pyramid, right? And we'll draw it pretty grossly here. So like a pyramid, we have an apex and a base. Well, the heart is no different. We have an apex, so the point of the pyramid is right there. And then we have the base. So the base of the heart is up here. Now we have to discuss the pericardium. This is our protection. So we recall back in AP1 we talked about all the serous membranes of the body. Well, this is one of them. Pericardium literally means around the heart. Recall that there are 2 layers, an innermost layer and an outer layer. The innermost layer being the visceral pericardium, so visceral layer. Then we have the outermost layer called the parietal pericardium. Now inside we have fluid. What does this fluid do? It acts as a shock absorber, so it is allowing for the heart to essentially sit into a liquid sac. It allowed the heart to beat unimpeded of causing friction or movement against the lung. So when that heart moves, if we didn't have this serous membrane, it would be rubbing up against the lungs constantly, causing friction, would eventually cook itself. So we have this layer, right? This membrane, again, protecting the heart, giving it a nice little area to beat friction free. So in elaboration, the pericardium consists of an outer fibrous pericardium and an inner serous pericardium. There's our 2 layers, the visceral and the parietal, and they're separated by a fluid-filled space. When we look at the arrangement of the heart and the cardiac muscle, we see that it is arranged in bundles. So these cardiac muscle bundles that form the myocardium. Again, myo meaning muscle, cardium meaning heart. Remember, we're going to look at the different layers of the heart. Now look at the muscular layer. We start with the first superficial bundles. The first superficial bundles form the atria. So we'll see these right here. Now from there we see that there are superficial bundles of the ventricles. So here we go in the outermost layer. And then if we were to cut a piece of those superficial ventricular bundles out, we get these deep muscles of the deep muscles within that ventricle. Now the arrangement, this is pretty interesting. The arrangement of the heart is kind of like a tornado. So if you envision a tornado, and how that tornado spins around and terminates at the point or the apex of that tornado. This is exactly how the heart works, twisting and turning and contracting in this cylindrical tornado-like fashion, moving blood throughout the body. Our next topic is going to be the layers of the heart wall. Now, this is all about nomenclature, and it's pretty straightforward. The heart has 3 layers, and they're named based on their location. So let's look at our prefixes. We see epi right here, then myo right here, and then ultimately endo. So we say, well, what exactly do these mean, right? So remember, epi means on top, myo means muscle, and endo means inside. So where are these layers? The epicardium, meaning heart, myocardium, and endocardium. So let's start with the outermost layer of the heart. So we take that visceral pericardium, and we see that the visceral pericardium is fused and continuous with the outermost layer of the heart. That would be the epicardium. And we see, we'll keep everything blue right here, we see the epicardium right here, right? And it is that visceral layer of the pericardium. Then we go to the thickest layer of the heart, okay? The thickest layer is the myocardium. So we see that myocardium right here. And if we were to bracket, right, maybe we'll change it to black so you can see a little better, bracket that area. So the muscle layer. Now this is probably the most important layer of the heart, because obviously this is what is contracting to help move the blood throughout the heart and also throughout the body. The innermost layer is known as the endocardium. So we see the endocardium right here. Now the function of the endocardium is actually pretty important. It has a specific texture or feel, more or less. Think of it like a slip and slide, okay? And I use this analogy to help explain why it is shaped or textured the way it is. So why is slip and slide? So if you've ever used a slip and slide, you know that in order to get that slip and slide to work, you need water, right? So think about what would happen if you tried to use the slip and slide if you had no water. And what we'll say is you are essentially the formed elements, the red blood cells, the white blood cells, the platelets, all the formed elements in blood. And the slip and slide is the endocardium. And now, if you, the blood, or the formed elements in the blood, tried to move through or slide down that slip and slide without the water, what would happen? You wouldn't go very far. You'd probably get some skin burns, etc. Well, the heart is no different. We need water. Where's the water? Well, what was 60% of blood, right? Whole blood. What was it? It was plasma. It was water. So that was the water needed for this endothelium, for this endocardium, for this slip and slide. So what it does is it produces a friction-free environment for the formed elements, for all that big stuff in blood, to kind of move through without damaging themselves, rupturing, forming clots and things like that. So it's super important that the inside of that heart layer is this smooth layer and resembling a slip and slide. Chambers of the heart. So we can take the heart and essentially split it up into different parts: upper halves, lower halves, left half, and right half. So why are we saying that? So let's take a pencil and kind of envision what we're doing here. We could turn it into upper halves and upper halves, lower halves, left and right halves. What are we talking about? Why are we dividing it this way? So what we're identifying are our chambers, our 4 chambers. The 2 atria, the upper halves are atria, and the 2 lower halves would be ventricles. So that gives us again 4 chambers, which would be separated by 4 valves. Now, what does this mean? Why do we do left and right? So basically what we're saying is, well, if we have these upper halves and these lower halves, each consisting of 2 chambers, why do we combine the upper and the lower to make a left half and a right half? What we're identifying is the side of the heart, the left versus the right, that is carrying deoxygenated blood versus oxygenated blood, respectively. We can look at the posterior view, finding and identifying some major structures like the right atrium and the left atrium, and the right ventricle and the left ventricle. So a couple things to also identify and note. So part of our coronary circulation. So we talk about circulation. We talk about the three different groups of circulation. One being pulmonary circulation, meaning blood going to and from the lungs, systemic circulation, blood going to and from the body, and coronary circulation, or the coronary circuit that is delivering blood to and from the heart. So what we see here is once blood has been used and abused, and we'll keep it blue to signify deoxygenated blood, once blood has been used and abused from the heart, all that blood is being returned to the actual right atrium via this coronary, right here, this coronary sinus. Now a couple other things to identify. Look at our typical rule. Remember we said that arteries are typically red and veins are typically blue. These are in diagrams. Again, remember we said that blood's always red, right? So this does not signify the color of the blood moving through it, just which is deoxygenated and which is oxygenated. So 9 times out of 10, you're going to always be right. If I say identify this red structure, you say artery, and I said identify this blue structure, you say vein. There's one place where that is going to be a little bit reversed, and that is right here in the pulmonary trunk. Okay, let's take a look at this pulmonary trunk. Our pulmonary veins are, look, red. And our pulmonary arteries are blue. So that's against our rule, right? So think about it. Why? So blood leaving the right ventricle is going to go to the lungs, and it's going to be a really, really high pressure. And because that pressure is high, we need the specific anatomy of arteries, these elastic arteries that's going to be able to recoil to that pressure. So we see that the pulmonary arteries are blue because they are carrying deoxygenated blood, but they need that anatomy, that elastic recoil, that elastic connective tissue to resist that pressure from the heart. But now once the blood is coming back from the lungs and entering the left atrium, the residual pressure is relatively low. So we convert over to veins. So again, changing the anatomy to account for the pressure differential. But that blood is now oxygenated. So we signify it as this red blood vessel. We have our aorta, okay, so our aorta, we'll put that red. Our aorta has a couple portions, the ascending aorta, the aortic arch, and then the descending, so blood leaves the heart, it goes up, around, and down to the body. We have 3 shunts off the aorta, a brachiocephalic trunk, and this is kind of getting ahead of ourselves, but look at our terminology, brachiocephalic, right? So what does this tell us? Brachial arm, cephalic head. So we're delivering blood to the head and arm. Then we have a left common carotid and our left subclavian. Okay. So major conducting veins of the heart are superior vena cava and our inferior vena cava. Okay. And we'll get into some of the smaller vasculature of the heart later on. So what we're going to do now is we're going to talk about the right atrium, the left atrium, the right ventricle, and the left ventricle, and some other structures in and around the heart. But we're going to do it in one slide. Now I know they use 4 slides in our presentation to discuss this, but we're going to kind of condense it all, look at one picture, and talk about it. Now, we already said the right atrium receives blood from the superior and inferior vena cava and the coronary sinus. So we have 3 dump-in points, okay? And we say dump-in points, meaning the places that the body is going to dump deoxygenated blood back into the heart. So let's find those right away. So here we go. We'll zoom on in. We'll use a pen to highlight, and I'll dash right here. This is the opening to the coronary sinus. So that back portion of the heart, that posterior view that we saw, that coronary sinus, that's where blood is coming in. And then coming down in through the superior and up through the inferior vena cava, blood is going to pool into this right atrium. Okay. Now, what happens? You need to be able to trace the flow of blood through the heart. So let's do the ventricles, the atria first, identify them. Then we'll talk about the valves, and then we'll talk about the flow of blood. So if this is our, we already said our, we'll highlight it in yellow. This is our right atrium. Okay, so we follow that right there. We then go to what chamber comes next in our series of events, and blood flows down, so we're hitting our right ventricle. So here it is, right ventricle. Now from the right ventricle, blood's going to move up to the lungs, come back, and now enter the, we'll zoom on out a little bit, into the left atrium. From the left atrium, blood's going to go down into the left ventricle, which is going to be the biggest of our chambers, the thickest, and we can see if we look at the thickness of this wall, it's about 4 to 5 times thicker than what's over here. You see highlighting a little bit in blue on the right side of the heart. So that left ventricle is so much bigger because it has to produce enough force to send blood in and out of the aorta and to the entire portions of the body. Now let's look at some structures. Let's start with the inside of the heart. We'll notice that it has a texture, right? And I call it Freddy Krueger face. This is, growing up in the 90s, you think of Nightmare on Elm Street. And if you've ever seen that movie, you know what Freddy Krueger's face looked like. We see all these bumps and grooves and streaks and texture. Now, this texture is called trabeculae carnii. Okay, now that is the texture inside the heart. Now, we can now look at the interventricular septum. So again, terminology, interventricular septum. It's what divides the left and right ventricles. We also have an interatrial septum. So if you were to kind of pinch behind these major blood vessels here into the left and right atrium, you'd be able to get that interatrial septum. So I'm going to go ahead and delete some stuff so that we can continue to discuss what's going on in this heart. So again, just to do some general anatomy, you have the apex at the point, the base at the top. We also are going to see a couple other things. Let's look at our valves. Our valves come in two varieties. They come in AV valves, which are known as atrioventricular valves, meaning they come between the atria and the ventricles. And we have semilunar valves, again, based on their shape, these half moon-shaped valves. And we have two of them, and they're named after where they go. So let's start with our AV valve. Our first AV valve, and we'll put it in red, is our tricuspid valve, and it separates the right atria from the right ventricle. Now we'll hit our second AV valve. It is known as the bicuspid or the mitral valve. Now again, let's look at the nomenclature. Tricuspid, 3 cusps to this valve. Now the bicuspid is much bigger. It is called the bicuspid because it looks like 2 valves. It's actually 4 valves that have been fused together to make 2 massive valves. Now, look what's attached to these valves. Strings. You ever heard that saying, don't tug on my heartstrings? Well, here are your heartstrings. They're called chordae tendineae. Now, these chordae tendineae, their job is to prevent backflow of blood into the atria when the atria are contracting or when the ventricles are contracting. So we'll talk about exactly how those work in a little bit because they work kind of counterintuitively. Then they're attached to muscles. So something's pulling on those muscles, and those are papillary muscles. So series of events, papillary muscles 1, chordae tendineae 2, bicuspid, tricuspid 3. It's only these AV valves that have the chordae. Our semilunar valves do not have chordae. They are pressure-driven valves. We have 2. The first being right here, since it goes to the pulmonary trunk, it's called the pulmonary semilunar valve. And we have our next one, which is our aortic semilunar valve. And again, these valves are leading to the lungs, pulmonary, and the aorta, aortic valve. Now, a couple other things to look at. Blood vessels. We have the pulmonary trunk. We already said the aorta. We have our superior and inferior vena cava, okay? We have our left and right pulmonary arteries, again in blue, and our left and right pulmonary veins in red. Now, a few things to talk about. I want to talk about fetal shunts. Now, we have a couple fetal shunts. In utero, we have 3 fetal shunts. It is known as the ductus arteriosus and the foramen ovale, and then our umbilical cord. In the heart, we have 2 of them. Now, upon birth, these holes close up. Now, again, they're shunts. The first is right here. We're going to zoom on into the right atrium. This shallow depression right here. Now, in utero, this was a hole. It was called, and we'll write it in, the foramen ovale. Now, what did it do? It connected the left and right atria, right? So it was a hole that connected the left and right atria, allowed blood to flow between the left and right atria. Now, upon birth, it closes up, called the fossa ovalis. The next one is up here. This in utero is called the ductus arteriosus. Now, why ductus? It was a duct that sent blood from this pulmonary trunk into the aorta. Again, another shunt. Now, at birth, it closes off and forms a ligamentum arteriosum. Again, this ligament that holds that aorta in with that high pressure. Now, why a duct? Why a hole? Why do we have these 2 fetal shunts? And we'll highlight them in green, right? So fossa ovalis and ligamentum arteriosum. They're bypasses. What are we bypassing? The lungs. In utero, we are in fluid. We cannot breathe in fluid, so we use these fetal shunts to bypass the lungs until birth when we're out in oxygen and able to use them appropriately. A review of the left atrium. So again, remember the previous slide, we went through all this anatomy. We continue with the left ventricle. Remember that it receives blood from the left atrium and then ultimately sends blood to the entire body, which is why that wall is 4 to 5 times thicker than the right ventricular wall. The fibrous skeleton. The fibrous skeleton of the heart serves several functions. It is a connective tissue frame which functions to electrically isolate the atria from the ventricles. The atrioventricular conducting system, which we're going to talk about a little later on in this lecture, is the only electrical connection between the atria and the ventricles, and again, this is in a normal heart. The fibrous skeleton also provides rigidity to prevent the dilation of individual valves or outflow tracts. Finally, it provides a point of attachment for our valves and those leaflets to the myocardium. The myocardium extends from the fibrous skeleton in a series of spirals that progress all the way to the point or the apex of the heart. Now, the skeleton consists of a set of fibrous rings of collagen, and it's going to surround the atrioventricular canals and extends to the origins of the aorta and the pulmonary trunk. Now, the interesting thing about the skeleton is that it lies within the plane of the base of the ventricles, which is roughly parallel to the coronary sinus. Heart valves and circulation of blood. Now, we identified anatomically which were atrioventricular valves and which were semilunar valves. So to reintroduce and reinforce, the valves of the heart open and close in response to pressure changes as the heart contracts and relaxes. Right and left AV valves, their job is to prevent backflow from the ventricles into the atria. This is known as retrograde blood flow. Right and left semilunar valves prevent backflow from the arteries into the ventricles. Now, we talked about the valves and the chordae tendineae and how it's counterintuitive to what you would think, right? So remember those papillary muscles. Here they are, papillary muscles. When they are contracted, people think that contraction of the papillary muscle means that the valves pull open. And this is where that counterintuitive notion lies. So actually, when the muscles are relaxed, the valves are open and blood is flowing from the atria into the ventricles. Now, on the flip side, when the muscles are contracting, they're pulling tight on the chordae tendineae, which essentially keeps the valves closed against the building pressure in the ventricles as it ejects blood out into the pulmonary and systemic circuits. Now the valves work opposite each other. So when one set of valves are open, the other set are closed. So for example, when blood's moving to the atria or to the ventricles from the atria, the AV valves are open and the semilunar valves are closed. Now, when blood's moving to the pulmonary circuit and the systemic circuit via those semilunar valves, both semilunar valves are open and the AV valves are closed. And we can see that in this picture. In this first one here, you can see the tricuspid and the bicuspid are open and the AV and the semilunar valves are closed. And in this picture here, the semilunar valves are open and the AV valves are closed. Our next job is to trace blood through the heart and through their respective systems and circuits. So we deal with systemic circulation and pulmonary circulation. Again, the systemic circuit is blood to and from the body, and the pulmonary circuit is blood to and from the lungs. So we have to pick a starting point. And whether we're talking about the electrical conductance through the heart or flow of blood through the heart, we're always going to start at the right atrium, right here, number 1. So what happens? The right atrium is going to contract. It's going to send blood into the right ventricle. The right ventricle contracts and is going to send blood through that pulmonary semilunar valve into the pulmonary trunk, and then it will diverge to the right and left lungs via the pulmonary arteries. Now the lungs are going to do their magic, so we're going to resort right to here. So the pulmonary capillaries is going to load up on oxygen and unload that CO2. And then that oxygenated blood is going to return back to the left and right pulmonary veins via the pulmonary veins into our left atrium. Now the left atrium is going to contract. It's going to pass through that bicuspid valve and enter into the left ventricle. Now that left ventricle, that real thick myocardium here, is going to build a ton of pressure, and then boom, it's going to hit that aortic semilunar valve, sending blood up the aorta and then into the aortic arch to the head, neck, arms of those 3 shunts right off the aortic arch, and then down. Will travel down that thoracic aorta into the abdominal aorta and then their respective distributing arteries. Now, once it hits those capillary beds in our tissues, we're down here now where we are unloading oxygen and loading up on carbon dioxide. Now from there, we're going to go back up the inferior vena cava, and then from the head, neck, and arms down into the superior vena cava and dump back into the right atrium. Now notice in our diagram over here we have the coronary sinus. So now we're kind of introducing that coronary circuit where blood from that's been used and abused by the actual heart muscle, the myocardium, is going to deliver back into the right atrium again, back to that starting point. Now you can click on the link to view an animation on coronary circulation. We will review it in the next slides, but remember you do have to be connected to the internet and in slideshow mode in order to run this animation. Coronary circulation. Blood's going to flow through coronary arteries, delivering oxygenated blood and nutrients to the myocardium. Now we're going to look at the branches that arise from the ascending aorta, the coronary arteries, and then how they exchange blood through their subsequent branches to the coronary veins, which are going to remove carbon dioxide and waste from the myocardium. The branches are going to converge at the coronary sinus, dumping blood back into the right atrium. So let's use a picture to identify these branches. So let's start with arterial circulation. So we see in the aorta 2 little thieves over here. These thieves are the left and right coronary artery. So we'll drop on out to see the right coronary artery. So the left and right coronary artery are going to take a little blood from the aorta as it comes out of the heart and deliver it to the myocardium. This is the beginning of our coronary circulation. Now, blood is going to flow, and we'll kind of highlight it in black here, through those left and right coronaries until we get to our branches. So on the right side, our first branch, or our branches that descend, known as our marginal branches. Okay, now we get 2 branches of bifurcation off the left coronary. The first that goes around to the back of the heart, this is known as the circumflex branch. We see the circumflex branch over here. And then we have the one that drops straight down to and in between the left and right ventricles, right over the interventricular septum. And it's known as the anterior, because it's in front, anterior interventricular artery. Now, clinically, this is also known as the left anterior descending artery, the LAD. Clinically, again, this is known as the widowmaker. The reason why it's known as the widowmaker is because blockage of this artery, if fully occluded, will cause what's known as a massive heart attack, essentially killing, depriving the left and right ventricles of blood, ultimately killing that myocardium, that heart muscle, a heart attack that a patient will not really recover from. So again, it's known as the widowmaker, and is also known as a massive heart attack. Now, the next thing we're going to talk about is from the posterior aspect. So anything that's really shaded here, shaded in light red, is the back view. Now, again, balance in biology, if there is an anterior interventricular branch, if we follow this right coronary artery around to the back, we lead to this branch back here. Again, this is the posterior view, and this is the posterior interventricular artery. Okay, so that pretty much takes care of the arteries, so pretty straightforward. Next are our veins. Now, our veins have a little different nomenclature, a little different wording. So let's start in front. Remember that anterior interventricular artery? Well, there's a vein that follows it. It's known as we'll highlight it in yellow here, as the great cardiac vein. Okay, so blood is going to be exchanged at the capillary network from the arteries. It's going to flow, remember, backwards. We're flowing up the veins around the back of the heart and converging onto the coronary sinus. So there's where we're pulling before blood, or that deoxygenated blood is going back into the right atrium. Now we have a couple other ones. Don't forget, if there was an anterior interventricular artery, there is a posterior interventricular artery, there is also going to be a vein that follows that posterior interventricular artery, but it has, again, a different name. We follow with gray down from that coronary sinus, and we have our middle. Well, actually, we're going up this way. We have our middle. So let's go with our pencil to show the flow of blood. We're moving up, around, and into. This is known as our middle cardiac vein. So let's highlight that in yellow, middle cardiac vein. Now we also have a small cardiac vein and an anterior cardiac vein. These are right here. And again, we follow this flow this way and around and to the coronary sinus. So again, this way, around, up, and to the coronary sinus. We can look at cardiac muscle, specifically the histology. Remember, similar to skeletal muscle, they have striations. They are involuntary, a lot of mitochondria, branching, single nuclei per branch, and more importantly, what differentiates them from skeletal muscle and smooth muscle are those intercalated discs. So let's go ahead and highlight those intercalated discs. Now, why are these important? These are really unique junctions. Their job is to link the cells together and define their borders, okay? So that's really important. But more importantly, they are major portals for the cardiac cell-to-cell communication, which is ultimately required for coordinated muscle contraction and maintenance of circulation through the heart and through the body. So again, we have these gap junctions that's going to regulate communication again with ions. So ion communication, specifically calcium, to spread across these muscle fibers, enacting that contraction phase. Let's look at the arrangement of the components in a cardiac muscle fiber. As you see, it looks strikingly similar to skeletal muscle. What are we missing? We're missing those terminal cisterns, storage tanks of calcium, but we do have all the basic same components: the transverse tubules, mitochondria. All of our protein filaments, actin, myosin, our M line, our Z disc to Z disc, our I bands, A bands, all the functional parts of the sarcomere. So in cardiac muscle cells, these individual cardiac muscle cells are joined together at their ends by those intercalated discs that we just talked about, forming these long fibers. And again, like skeletal muscle, each cell is going to contain those myofibrils, those specialized protein fibers that slide. Remember that sliding filament theory that we talked about? That slide past each other. These are going to be organized into this functional unit, the sarcomere. Okay? And it is the fundamental contractile units of the cardiac muscle cell. So we're going to continue with the heart by looking at the how. How does the heart contract? How does it beat? And this brings us to the conduction system of the heart. Now there are cardiac muscle cells, the ones we've been talking about. They are self-excitable and therefore autorhythmic. So that's important, that autorhythmicity of the heart. Now, these cardiac muscle cells repeatedly generate spontaneous action potentials. And these spontaneous action potentials are going to trigger the heart contractions. Now, the cells form the conduction system, which is the route for propagating action potentials through the heart muscles. So what we're going to do is look at that point from threshold, the depolarization and the repolarization event that occurs, and we're going to call those graded potentials, those pacemaker potentials, the part that's going to cause and drive that autorhythmicity. So our job now is going to be to identify the parts of the conduction system and then elaborate on it. So again, where do we start? We're starting at the right atrium, and we start right here at the SA node, the sinoatrial node. It's also known as the pacemaker of the heart. This is the place where we are setting the pace, setting the way, the beat, the time that the heart is going to either increase or decrease its contraction rate. So again, if you think about exercising, the sympathetic nervous system is going to activate the pacemaker of the heart and increase those pacemaker potentials, essentially making the heart beat faster. And then on the flip side, when we're resting or sleeping, the parasympathetic nervous system will activate the SA node by suppressing it, decreasing those pacemaker potentials, decreasing action potentials, and allowing the heart rate to decrease. So from the SA node, action potentials are going to spread across the atria and then activate the second part of this step, which is the AV node, which is in the floor of the right atrium and then into the top of the right ventricle. From there, action potentials travel down the bundle of His, which is also known as the atrioventricular bundle, and then it bifurcates, it branches. Okay? These branches are called left and right bundle branches. Now, from here, we're going to reach the apex of the heart and we're going to branch up. Now, we're going to branch up into the papillary muscles that connect to the chordae tendineae and then the valves, and then into the myocardial wall of the ventricles. These are called Purkinje fibers. Okay, so in our next slide, we're going to put it to time, how long it takes to run an entire cycle, and how these steps 1 through 5 work together to get one heart cycle, heart contraction cycle. So we can look at influences on the conduction system. So we've alluded to the sympathetic and parasympathetic system. We haven't really introduced any hormones, but the autorhythmic fibers in the SA node are the natural pacemaker of the heart because they initiate action potentials most frequently. Signals from the nervous system, here we are with the parasympathetic nervous system and sympathetic nervous system, and hormones like epinephrine, adrenaline, that make your heart rate increase. These all can modify the heart rate and force of contraction, but they do not set the fundamental rhythm. So that's important. They modify the rhythm but do not set it. Let's elaborate on that impulse conduction through the heart. Let's put it to step with a little more detail and to time. So at time 0, step 1, the SA node activity and atrial activation begins. So we're firing that first action potential. Step 2, those action potentials, that stimulus spreads across the atrial surfaces and reaches the SA node. This takes about 50 milliseconds. And we see now for step 3 in purple, all the atria, both atria are fully depolarized. There's a 100 millisecond delay at the AV node, and atrial contraction begins. So this takes about another 100 milliseconds, total elapsed time about 150 milliseconds. Now blood is moving. It's moving from the atria into the ventricles. Now step 4, the impulse travels along the interventricular septum within the AV bundle and the bundle of His to those Purkinje fibers in the ventricular wall at the apex and into those papillary muscles. And they move via the moderator band to the papillary muscles of the right ventricle. And again, this takes about 25 milliseconds, so total elapsed time is about 175 milliseconds. Now, those impulses, that action potential spread and is distributed by the Purkinje fibers and relayed throughout the ventricular myocardium. So now both ventricles are fully depolarized and contraction will begin. Now, atrial contraction is completed, meaning all blood is now in the ventricles, and the ventricular contraction begins. It takes about 50 milliseconds for those ventricles to completely depolarize. Right? Our total elapsed time is 225 milliseconds. And at the end of step 5, blood is moving out of those semilunar valves to the lungs and to the body. Let's take a look at those action potentials in a ventricular fiber. Now, it looks a little different than our muscle fibers. An action potential in a ventricular contractile fiber is characterized by a rapid depolarization. So that's step 1. And that's good because once we hit threshold, we rise quickly up to plus 20. Okay? And this is due to those voltage-gated fast sodium channels opening. Then we get this plateau phase. Now that plateau phase is important. We're maintaining that depolarization because we're allowing calcium inflow when these voltage-gated slow calcium channels open, and the potassium outflow channels are opening as well. So what's this plateau phase for? It's occurring to allow and sustaining a longer contraction cycling of the heart muscle. This allows time for blood to essentially move to its respective parts. Now, why is this important? It's again important because we don't want an abnormal rhythm or an arrhythmia. This is so that blood has time to move. So we can look at clinical examples. Clinical example would be atrial fibrillation, where the atria, or even ventricular fibrillation, where the ventricles are quivering instead of allowing for this consistent elongated contraction cycle. And that quivering, which would look like a regular skeletal muscle twitch, does not allow blood to move. Instead, blood stays pretty stagnant, and this would be detrimental to life and would cause death. So we need this plateau phase, and again, it's because of that inflow of calcium that is keeping the cell depolarized. Now, towards the end of this plateau phase, calcium channels close, and then outflow of potassium will increase, bringing us to the repolarization. But before we get into the repolarization, let's talk about this phase right here. This is very important. We'll make it black. This refractory phase. So we saw this before in muscle, this absolute refractory. Now this is occurring during that plateau phase and parts of the repolarization phase. Why? What's happening? There cannot be another action potential coming to stimulate another depolarization phase until this contraction is complete. And that's really important because there again comes our abnormal rhythmias or arrhythmias, okay? Now, what happens in step 3? The closure of those calcium channels and opening of the potassium channels. So outflow of potassium when additional voltage-gated potassium channels open brings us back down to resting membrane potential. Now, once we're back down into our RMP range, we are into a relative refractory period where, yes, another action potential can come and stimulate another cycle up, down, and across and down. Perfect. ATP production in cardiac muscle. Now, cardiac cells contain numerous mitochondria which enable continuous aerobic respiration and production of ATP for cardiac function. Now, the myocardium requires significant energy to contract continually over the human lifetime. Remember, the heart is one of the only muscles in the body that is continually contracting. So these energy needs are met through mitochondria, myoglobins, and rich blood supply from the coronary arteries. Now, the mitochondria generate ATP for the contraction of the cardiomyocytes. Myoglobins are oxygen storing and oxygen transferring pigments in those cardiomyocytes. Aerobic metabolism occurs when oxygen is present, while anaerobic respiration occurs when tissue is deprived of oxygen. Aerobic metabolism accounts for nearly all of the metabolic function in the heart, but anaerobic metabolism may contribute as well. Now, we do have glucose reservoirs and lactate recycling, which allows the heart to function even during malnutrition. So again, remember what lactate is. Lactate is a molecule that's produced by anaerobic respiration that can be used to produce ATP without oxygen, albeit this is at lower levels. Now let's look at myoglobin. It is a smaller globular protein containing one heme group that carries oxygen to muscles from the blood and stores reserve oxygen. This is different from hemoglobin, which has 4 heme groups. There's only one heme group. Now, the interesting note, again, the heart muscle pumps continuously throughout the life and is adapted to be highly resistant to fatigue. Those cardiomyocytes contain large numbers of mitochondria, which are, again, those powerhouses of the cell, enabling continuous aerobic respiration and ATP production required for mechanical muscle contraction. Now, it's funny because we talk about how energy-hungry cardiac muscle is. It is one of the highest required muscles for energy in the entire human body, second to the brain, and has a high level of mitochondria and a constant rich blood supply to support its metabolic activity. Let's elaborate on the aerobic metabolism. Now, the heart derives energy from aerobic metabolism through different types of nutrients. 60% of the energy used to power the heart comes from fat, freeing fatty acids and triglycerides. 35% from carbohydrates, and 5% from amino acids, and ketone bodies from proteins. The proportions do vary widely with available dietary nutrients. Now, malnutrition will not result in the death of the heart tissue in the way that oxygen deficiency will, because the body has plenty of glucose reserves that sustain all these vital organs and the body, and the ability to recycle and use lactate aerobically. Heart muscles also contain large amounts of that pigment that we call myoglobin. Again, remember, it's similar to hemoglobin, except it only binds one oxygen. It transfers the oxygen from the blood to the muscle and stores that reserve oxygen for aerobic metabolic function in the muscle cell. Now let's talk about anaerobic metabolism. Now, while aerobic respiration supports normal heart activity, we do rely on anaerobic respiration to provide additional energy during brief periods of oxygen deprivation. Lactate, which is created from lactic acid fermentation, accounts for the anaerobic component of cardiac metabolism. At normal metabolic rates, about 1% of energy is derived from lactate, and then about another 10% under moderately hypoxic, again remember hypoxia is low oxygen conditions. Under more severe hypoxic conditions, not enough energy can be liberated by lactate production to sustain ventricular contraction, and then ultimately the heart will fail or heart failure will occur. Now, lactate can be recycled by the heart and provides additional support during nutrient deprivation. Recycling lactate is very energy efficient in the nutrient deprived myocardium, since one NAD plus is reduced to NADH and a hydrogen ion, and this equates to about 2.5 to 3 ATPs being liberated. Now this, when lactate is oxidized, it produces pyruvate. The produced pyruvate can then be burned aerobically in the citric acid cycle, liberating a significant amount of energy. Now, creatine phosphate, this is important because we do store creatine phosphate in our heart muscles as a last-ditch source of energy. Now, during times of myocardial infarction or heart attack, an important note, if you have a small heart attack or a minor heart attack, not a major infarction, what your doctor will do is take blood and test that blood for creatine kinase. Now remember, creatine kinase is an enzyme found in the heart, brain, skeletal muscle, and other tissues. If we increase the amount of creatine kinase in the blood, that means that there were heart muscle or muscle tissue that has been damaged. So the tests that measure this can indicate a heart attack has occurred. So how do we measure that electrical conductance through the heart? Well, we use an EKG or ECG, an electrocardiogram. Now, the electrocardiogram is used to measure those various steps, those 5 steps, for the electrical conductance through the heart. Now, we can identify peaks and segments and what they mean. So for us, we're really going to look at what this P wave represents, the QRS complex, and the T wave. We're not going to get too much into the intervals and the segments. So let's start with the P wave. The P wave is equal to atrial depolarization, right? So what does that mean? That means that the atria are contracting. The QRS complex equals ventricular dipole. Okay, so the ventricles are contracting. The T wave equals ventricular repolarization, right? So now the ventricles are relaxing, they are regaining and coming back to resting membrane potential, ready to start another cycle. So this is one cycle, right? So we look at a subsequent PQRST wave, another complex, and we can measure from R to R. So let's assume that we have another R over here, and this R to R equals heart rate, okay? Now there's a quick and dirty way to calculate heart rate by looking at an EKG. We can count these large boxes between successive R to Rs and take 300 and divide by, if we say there was 3 boxes between R to R, the heart rate would be 100 beats per minute, okay? Now, one last thing. Everything should be paired, right? We have atrial depolarization, ventricular depolarization, ventricular repolarization. Where is, so let's write in, where is atrial dipole, right? Where is this? It should be here. It is a phenomenon that occurs. How come we don't see it? So the answer is pretty simple, right? If you think about it, it should come before the T wave. So it should be somewhere, and we'll highlight where it should be, a little region. It should be somewhere here. So how come we don't see it? Well, that QRS complex is so big that it tends to mask. So we're going to say it's masked. Tends to mask the atrial dipole graph. So that's why we don't see it. Let's start overlapping cycles. So action potential propagation through the heart in conjunction with the EKG. So what's happening? Number 1, as that P wave is going up, our action potential in the SA node, depolarization of the atrial contractile fibers produce that P wave. Then we have atrial systole. So remember, systole is contraction. Okay, so the atria are contracting at the time period that goes from the end of the P wave all the way to the QRS complex. So step 3, depolarization of the ventricular contractile fibers produce that QRS complex. As a result, ventricular systole ensues. So after the QRS, the ventricles contract, and ventricular systole goes in that time period, that interval from the end of the QRS complex all the way to the T wave. Now for the T wave, we get repolarization of ventricular contractile fibers, again producing that T wave, and then we get into ventricular diastole. So we have introduced some new words, systole and diastole. Diastole meaning relaxation, systole meaning contraction. Now there is also atrial systole, but because those atrial repolarization chart or graph is masked, we do not see it in this 6-step process. The cardiac cycle. So one entire cardiac cycle consists of the contraction, so systole, and relaxation, diastole, of both atria, rapidly followed by the systole and diastole of both ventricles. Now there are a couple things that are occurring here. One, the electrical events that we covered in the propagation of action potentials through the heart. Pressure changes, so what's occurring in the ventricles and the atria as they are filling and emptying. Heart sounds, which is called auscultation, so we'll write auscultations. So listening to the heart sounds. The volume changes that occur and the mechanical events that lead to these volume changes, then the heart sounds as well as the pressure changes. So we're going to look at each of these portions individually and then overlap them to see how they work together to contribute to the cardiac cycle. So a summation of our EKG, our electrical events, so from middle of P to the right before the end of QRS, we have atrial systole. From S to through our T wave, we have ventricular systole. Then we have our relaxation period, which lasts about 0.4 seconds. So one cycle, we're looking at about 0.5 seconds. Let's take a look at pressure. So a couple things to note. Our pressure runs from a scale of 0 to 120. Now, if you have ever had your blood pressure taken, you know that a typical blood pressure rate is systolic over diastolic is about 120 over 80. So remember those numbers because here we are, that 120 and that 80. Remember, we're reading on a systemic level, we're reading off that brachial artery. So we're talking about pressure that comes out of the aorta. So let's start there. Let's look at aortic pressure. So during atrial systole, so the atria are contracting, pressure within the ventricle, so after the atria contract, look at pressure. It goes from 80 all the way up. We'll change over to black. We'll go all the way up to 120. Okay? And then as the ventricles contract, the pressure in the ventricles decrease back down to that 80 mark until we get back into aortic pressure. But then we get this weird dichrotic wave, and we have to explain what this is. It is a small and brief increase in arterial pressure. So see how it goes down and back up and back down. What this is due to, it's due to the closure of the aortic valve. So what happens is that we get this short period of retrograde flow of blood immediately before the aortic valve closes, and that backflow of blood causes a spike in pressure during that relaxation period back into the ventricles, back into the aorta. Now, that takes care of our aortic pressure. And again, we can measure this when you put on a blood pressure cuff, and that's why we see these ranges of 120 to 100. Now, we can look at our blue line, right? So what happens when the aortic valves open and close? We can talk about what happens when the bicuspid valves open and close. So let's follow through with our blue line, okay? Now, this blue line represents ventricular pressure. So as blood moves from the atria, so we have atrial contraction, ventricular pressure starts to rise, and it rises all the way up to 120 millimeters of mercury. Again, we're in the left ventricle. So that pressure has to be relatively high. So that ventricle is going to contract. It's going to push blood into and towards the lungs and the rest of the body until we get back to atrial systole. So back to filling, back to increase in pressure due to the atria filling and then subsequently pushing them into the ventricles. The last one we're going to look at is atrial pressure. Now remember, the atria don't produce that much pressure because they're not going very far. It's only pushing blood to the ventricles below it. So we get atrial systole, so pressure builds within the atria as it contracts. Once it contracts, it ejects blood into the ventricles, causes an increase in pressure, and then we stabilize again until we get our relaxation period, and then we go back into our left atria into the left ventricle again. Cardiac auscultation, or heart sounds. Now, all these carefully coordinated sequences of events that we've been talking about or describing so far, again, in a healthy heart, blood flows smoothly, and so it cannot be heard on auscultation. So when you put that stethoscope towards the heart, you're not hearing blood flowing. Blood flow is smooth. What can be heard, though, is these two sounds. We describe them as a lub and dub. The sounds you are hearing are the vibrations caused by the blood, the turbulence of the blood against the closure of the valves in the cardiac cycle. So we have S1, our first lub, occurs at the end of the filling phase when the atrioventricular valves snap shut. Our S2 is the dub. This occurs at the end of the outflow phase when the outflow valves snap shut. Now, damage to the heart valves can cause them to become narrowed, this is called stenosis, or leaky, which is called regurgitation, allowing for retrograde flow. Either of these conditions can lead to turbulent blood flow, which is heard as a humming sound on auscultation of the heart. These sounds are known as murmurs. Now, occasionally, with clinical practice, so if you practice enough with your stethoscope, we can hear additional sounds. These sounds are termed S3 and S4 and can typically be heard during certain situations. So S3, this sound is sometimes heard early and diastole following S2. In young people or athletes, it is normal. However, in older people or patients, it often indicates congestive heart failure. What causes it is the deceleration of blood moving from the left atrium to the left ventricle. So that's S3. Now S4 can be heard sometimes during atrial contraction. This is late diastole, immediately before S1. And it's associated with reduced ventricular compliance. So typically as we get older, our ventricles become stiff. So this would lead to reduced compliance or even left ventricular hypertrophy or ventricular growth. Now, where do we place the stethoscope to hear these sounds? So for the aortic area, we're going to place our diaphragm of the stethoscope at the second intercostal space. Now these are spaces that you can palpate with your fingers. And at the right sternal edge. This is the anatomical landmark for the aortic valve. You could listen for at least 5 seconds for the second heart sound, which represents the aortic valve closing. Now for the pulmonic area or the pulmonary area, we're going to place our diaphragm of the stethoscope at the second intercostal space and our left sternal edge. This is the anatomical landmark for the pulmonary valve. Again, we're going to listen for about 5 seconds for the second heart sound, which represents the pulmonary valve closing. For the tricuspid area, again, place your diaphragm of your stethoscope at the 4th to 5th intercostal space and left sternal edge. This, we could listen for, again, 5 seconds for the first heart sound, which is going to be our tricuspid valve closing. The fourth location for the mitral area would be to place your diaphragm at the 5th intercostal space, mid-clavicular line, which would be the same area as the apex beat. We listen for 5 seconds for the first heart sound, which represents the mitral valve closing. Let's now take a look at volume within the ventricles. Now we can calculate stroke volume. Okay, and what stroke volume is the amount of blood that's going to be ejected through the heart per beat. Okay, now we can calculate this. So let's look. The atria contracts, starts filling the ventricles are going to contract. What we can take is we could take end diastolic volume, which is 130 milliliters, subtract it by end systolic volume, which is roughly 60, which gives us an average stroke volume of about 70 mils. So this is our stroke volume. So an average healthy male will eject about 70 milliliters of blood per contraction, per beat. Let's take a look at some of these mechanical events. So the cardiac cycle, remember, has and can be roughly divided into 4 stages. Our filling phases, so here's our filling phases, okay? An isovolumetric contraction, which we'll talk about in a little bit. An outflow phase, so an ejection phase, and an isovolumetric relaxation phase. Now the filling phase is when the ventricles fill during diastole and atrial systole. Our isovolumetric contraction phase is the ventricles contract, building up pressure, which is allowing for this system to be ready to pump blood into the aorta or the pulmonary trunk. Our outflow phase are the ventricles continue to contract, pushing blood into the aorta and that pulmonary trunk. And this is again also known as systole. So we see systole right here during our outflow or ejection phase. Now, isovolumetric relaxation, this is a pretty interesting point. The ventricles relax. They are ready to refill with blood in the next phase. Now, why is this different from isovolumetric contraction and isovolumetric relaxation? So, in the contraction phase, what's happening is pressure's building. It's building, 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 building. Isovolumetric means no movement. So, as pressure builds within the ventricles, blood's going nowhere. Look at the mechanical events. All the valves are closed. So because all the valves are closed, pressure is building until we reach threshold and forces those semilunar valves to open. So let's look and elaborate on the filling phase. So the ventricles are filled with blood in two stages: diastole, which is heart relaxation, and atrial systole, which is contraction of the atria. In diastole, both the atria and the ventricles are relaxed. Now, blood is going to flow from the vena cava and pulmonary veins into the right and left atria, respectively. Now, before flowing directly into the ventricles will fill with blood at a steadily decreasing rate until the pressure in the ventricles is about equal to that of the veins. At the end of diastole, the atria contract. This squirts a small amount of extra blood into the ventricles. So this increases the pressure in the ventricles so that it is now higher than it is in the atria, causing those AV valves to close. Now, that brings us to isovolumetric contraction. This is one of my favorite topics, just because it's so interesting. Now, as contraction begins, both sets of valves are closed, meaning that no blood can escape from the ventricles or is moving through the heart. The start of systole, therefore, massively increases the pressure within the ventricles, ready to eject blood into the aorta and pulmonary trunk. So the stage of isovolumetric contraction lasts for about 50 milliseconds, where pressure is building within the heart, all the valves are closed, and no blood is moving. That leads us to ventricular ejection or our outflow phase. Once the pressure in the ventricles exceeds the pressure in the aorta or pulmonary trunk, the outflow valves, meaning the aortic and pulmonary semilunar valves, open. Blood is pumped from the heart into the great arteries of the body. Now, at the end of systole, it takes about 330 milliseconds later, the ventricles begin to relax, and it's going to decrease the pressure compared to the aorta, which causes the valves to close. As well as this, blood begins to flow backwards through the outflow valves, so the semilunar valves, which also contributes to the closure of those valves. That brings us to isovolumetric relaxation. Now, at the end of the ejection phase, both sets of valves are closed once again. The ventricles are now going to start relaxing, reducing the pressure in the ventricles so that the AV valves open. The ventricles then begin to fill with blood, and the cycle begins again. So one of our last slides for looking at all these events is to superimpose them and kind of see, with regards to electrical events, pressure events, heart sounds, volume in our ventricles, and those phases in the cardiac cycle, what's occurring when you put everything together and look at the entire picture. So this is important. So an example would be during the QRS complex, we see that we're in atrial systole. We can correlate to a pressure sound as far as the beginning of our S1 sound, ending with end diastolic volume, and discussing our atrial contraction here. And again, just follow that track all the way down, and we can overlap and discuss what's occurring on all these phases. So this is your job, is to take each of these individually and kind of superimpose them and see what's going on during ventricular ejection, or even during this small period. Look how small the isovolumetric contraction is. We can follow that up. We see we're at EDV and diastolic volume, right into S1, ending with the little increase in pressure of the left atria right here. And then going in where the aortic valves open, and then right at the bottom of that S wave of the QRS complex. So we've talked about stroke volume. Now we're going to introduce cardiac output. So cardiac output is the volume of blood that's ejected from the left or right ventricle in the aorta or pulmonary, into the aorta or pulmonary trunk each minute. So stroke volume is the amount of blood ejected per beat, this is how much blood is ejected per minute. So this should be a pretty simple calculation. So cardiac output is a mil per minute figure. So we take stroke volume, multiply it by heart rate, and we could run this pretty simply, right? We said before that average stroke volume is about 70 mils, and we'll say that heart rate for a healthy individual, let's put it at 70 beats per minute. So that gives us a cardiac output, if you're ready for this number, 4,900 mils per minute. So that means that every minute your heart is beating 4.9 liters of blood. That's amazing to think about it. Talk about the workhorse of your body. Your heart is pushing out about 5 liters of blood every minute. Now look, if your heart rate, say you're not as athletic, and your heart needs to work harder, heart rate increases. Let's say your resting heart rate is 100 beats per minute. Your body is now kicking out 7 liters or 7 mils per minute. Now, what if you're athletic and your heart rate's more efficient? That means your cardiac output goes out. So we can take 70, multiply it by 60, and then we can say our cardiac output is 4,200 mils per minute. And ultimately this tells us, look, our heart is more efficient at distributing oxygenated blood to the body, so we have to beat less, which means our cardiac output is ultimately less, about 4,200 mils per minute. So what regulates and affects stroke volume? So we talk about regulation of stroke volume being a 3-factor system. So 3 factors regulate stroke volume. 1, preload, so we'll write a 1 here. 2, contractility, and 3, afterload. So contractility is pretty straightforward, right? The ability to which the heart is able to contract and the ease at which it's able to comply. Now, we already introduced a few slides ago that as we get older, compliance of the heart reduces or gets worse. Our heart becomes harder or even hypertrophic, so it becomes less compliant, harder to work, which means that our stroke volume is going to be affected. Now let's look at preload. Preload is, and I like this little diagram here because it kind of shows the heart and shows what happens before and after, right? Pre and after, preload and afterload. So preload is the volume of blood in the ventricles at the end of diastole, so end diastolic pressure. So it's increased in hypervolemia, so more volume of blood, so the volume of blood increases. It's increased in regurgitation of cardiac valves, and it's also increased in heart failure, so pushing more heart into the blood to try to get our afterload increased. Now, what is afterload? Right? So afterload is the amount of blood that's leaving the heart, but this needs to increase based on a couple of things like resistance. So for afterload resistance, the left ventricle must overcome to circulate blood. So we talk about resistance being length of blood vessels, the diameter of the lumen of the blood vessels. These are all things that put a clamp, and that's why I like this picture. Puts a clamp on the blood vessel. So the bigger, the heavier, the more hard the clamp, the more afterload, the more pressure the left ventricle has to exert in order to overcome that to distribute blood. So what increases afterload? Things like hypertension, high blood pressure, vasoconstriction, right? That clamp down on that artery, make the lumen smaller. We're increasing blood pressure. So increased afterload equals an increase in cardiac workload. That makes sense, right? That's why hypertension is so bad. Your heart's working overtime. It's working harder. It's trying to produce more pressure. Now, what does that mean? Your nice arteries, your aorta, that delicate aorta that's made up of elastic fibers, it can rupture. Right? It's called an aortic aneurysm. Think about, for you girls out there, your ladies, you use hair bands. Are they good forever? And the answer is no. The more you use them, the more you stretch them, eventually they get brittle, weak, and they break. Well, your arteries are no different. So exerting a high pressure for a long period of time is ultimately really bad for your arteries and lead to clinical conditions like aortic aneurysms. Next is regulation of heart rate. There are several factors that regulate heart rate. Number 1, the autonomic nervous system. We said fight or flight, increase in heart rate. We talked about parasympathetic, rest and digest, repair, decreasing heart rate. Hormones, adrenaline, part of that fight or flight response, can increase heart rate. Certain ions, like calcium. So calcium ion level have great impact on both heart rate and contractility. So as the levels of calcium ions increase, so does heart rate and contractility. So these high levels of calcium ions is called hypercalcemia, can be implicated in those short Q2 intervals and a widened T wave when we look at that EKG. And this ultimately increases heart rate. On the flip side, we look at sodium. Sodium has to be intricately and exactly regulated in the heart, right? So the flow of sodium into the muscle cells, if it's disrupted, the heart does not beat regularly. So it's important that we have appropriate levels of sodium and that we're tightly regulating the amount in or out. If we don't get enough sodium into the heart, we can disrupt heart contraction and heart function. Age. The older we get, the harder our heart has to work to pump. Children have a faster heart rate. And this is because their metabolism is so much faster. So till the age of 2, we see a really fast metabolism and a really fast heart rate. Once children reach those teenage years, their heart rate kind of slows down and is equivalent to that of an adult. Now, gender. Females typically have a faster heart rate than males, and it's suspected that female hormones and increase of estrogen and progesterone have a play in this. Physical fitness. The more physically fit you are, the better your heart is adapted to pump blood, so it beats at a slower rate. So if you look at the heart rate of an athlete, maybe 55 to 65 beats per minute. To a person who lives a sedentary lifestyle, maybe 75 to 85 beats per minute. Temperature. Temperature plays a role in heart rate. The warmer the weather, the easier it is for your blood to flow, the less your heart has to work. Nervous system regulation of the heart rate. So our input to the cardiovascular center comes from higher brain centers, the cerebral cortex, the limbic system, and the hypothalamus. From our sensory receptors, like our proprioceptors that are monitoring movements, our chemoreceptors, which is monitoring the blood chemistry, and our baroreceptors that are monitoring blood pressure. Now the output to the heart comes from our increased rate of spontaneous depolarization in that SA node and subsequently the AV node, which ultimately increases heart rate. This is part of our sympathetic nervous system, our cardiac acceleratory center. This also increases contractility of the atria and ventricles, ultimately increasing stroke volume. We have our vagus nerve, cranial nerve 10, one of the most important cranial nerves in our body, which is part of our parasympathetic division, which is going to ultimately decrease the rate of spontaneous depolarization in the SA node and subsequently the AV node, ultimately decreasing heart rate. Let's look at all the factors that increase the cardiac output. So let's start up here. Increased end diastolic volume. So increased end diastolic volume stretches the heart. This ultimately increases preload. Within the limits, those cardiac muscle fibers contract more forcibly. With stretching, this follows the Frank-Starling law of the heart, increases stroke volume, increases cardiac output. Positive ionotropic agents such as increased sympathetic stimulation, like our adrenaline, catecholamines, glucagon, or thyroid hormones in the blood. Here we go with increased metabolism. Increased calcium in the extracellular fluid. Increased contractility. So those positive inotropic agents increase force of contraction at all physiological levels of stretch, increase stroke volume, increase cardiac output. Decreased arterial blood pressure during diastole, decreases afterload, so factors that decrease afterload. The semilunar valves open sooner when blood pressure in the aorta and pulmonary artery is lower. It ultimately increases stroke volume, increases cardiac output. Let's look at the nervous system. Cardiovascular centers in the medulla oblongata receive input from the cerebral cortex. So there's that medulla oblongata again. Remember we said that the medulla oblongata was our go-to center? Well, here it is. The limbic system, proprioceptors, baroreceptors, and chemoreceptors increases sympathetic stimulation and decreases parasympathetic stimulation, fight or flight. Increase heart rate, increase cardiac output. Catecholamines or thyroid hormones in the blood, moderate increase in extracellular fluid. So these chemicals increase heart rate, increase cardiac output. Other factors, infants, senior citizens, females, higher heart rate. Low physical fitness, higher heart rate. Increased body temperature, increases heart rate, increase cardiac output. Exercise and the heart. It is important. Your heart is a muscle. You need to exercise it. Regular aerobic exercise can and will increase cardiac output, making your heart more efficient. Increase high-density lipoproteins, HDL. This is the good cholesterol. Decrease triglycerides. This is circulating fat. Improve lung function, decrease blood pressure, and assist in weight control. So again, what we're doing is decreasing our chance for buildup of atherosclerotic plaque. Sclerotic plaque buildup in our arteries. So we have some disorders that disturb homeostasis. These include coronary artery disease, atherosclerotic plaque buildup, congenital heart defects, arrhythmias, and congestive heart failure, all of which will increase your chance of myocardial infarction, so myocardial infarction or heart attack.